Today we will be talking about antibodies, immunoglobulins, right? The concepts about immunoglobulins are very important. These are one of the most important molecules in our body. We cannot survive without them in such a hostile world where there are so many microbes and so many pathogens which are trying to invade our body and destroy our tissue. Now, exactly what are antibodies? Antibodies. They are also called immunoglobulins. Now, what are exactly antibodies? From where they come in our body and exactly where they are present, right? Major portion of the antibodies is circulating in the blood, right? Antibodies are special protein molecules. Antibodies are very special type of protein molecules which are secreted by plasma cells. Antibodies are special type of protein molecules which are secreted by the plasma cells. Now, what is a plasma cell? Actually, whenever a B lymphocyte becomes fully differentiated and becomes fully functional, right, it starts producing antibodies. It starts secreting Y-shaped molecules or antibodies. So, antibodies are the products of proteinaceous product of well differentiated B cells or protein products of plasma cells. Is that right? Now, exactly in the blood, in which component of the blood antibodies are present? Yes, please. Question goes to very impressive Dr. Safa. In which component of the blood antibodies are present? Let's come with very simple thing. Everyone knows that blood has two components, cell and, yes please, plasma, and plasma has many components. Is that right? One of the component is plasma proteins. Actually, antibodies are the part of the plasma proteins. It's a very basic concept that where antibodies are. Antibodies are present, yes, present in your blood as the proteinaceous component of the plasma. Now, plasma has many types of proteins. Now, we have to see that within the plasma and within the plasma protein where antibodies are exactly present. So, Dr. Israel may answer us that your plasma has proteins and one component of the plasma proteins are basically antibodies. Now, which component of the plasma protein is having antibodies? I'm about to be impressed by him. I think he is very serious right now. He doesn't want to answer. So, I will do it myself. Look, one of the way, let's suppose that on this filter paper, we put a drop of plasma, right? We put all the plasma proteins here. We put all the plasma proteins here. You know, plasma proteins are positively charged or negatively charged? You like to keep secrets, you don't tell me. Plasma proteins are negatively charged. Is that right? At body pH, which is 7.4 by the way, at the body pH, plasma proteins are negatively charged. Now, because most of the plasma proteins are negatively charged, if you put a drop of plasma on this filter paper and you run the current across it, for example, you put negative electrode here and positive electrode here, of course, plasma proteins will move from negative electrode towards positive electrode because plasma proteins are themselves negative. So, they will be repelled by the negative and of course, they will be attracted to the positive. So, plasma protein will start moving. Now, as plasma proteins start moving, they segregate into group because all the plasma proteins don't move, right, with the same speed on this paper. You can answer this. If plasma protein is very heavy, right, that will move fastly or slowly? Slowly. Actually, those plasma proteins which are more negative and smaller in molecular weight, they will be more smart and they will rush fastly. So, what really happens that some of the plasma proteins will reach here, right? Then there will be some here here and here and here. Now, these are the plasma proteins which are smallest in molecular weight, right? And they are more mobile, right? So, they will come over here. This was the movement, right? Heavier plasma proteins will be left behind. Is that right? If you divide the plasma protein or segregate the plasma protein by this way, this is called process of electrophoresis. Have you heard of it? Process of electrophoresis. When you do electrophoresis of the plasma proteins, right? The electrophoresis. Is that right? Now, electrophoresis, when you do, the plasma proteins, the smallest molecular weight will be here, and as the plasma proteins become more and more heavier, they are segregated in a different band, right? Actually, these plasma proteins as a group are called, just please, albumins. Because albumins are the smallest molecular weight. And this whole group of plasma proteins, this is called globulins. Globulins. And this is post-fibrinogen. Is that right? Another way to show this is that you draw a graph. Graph is that if you put molecular weight here and you are classifying the plasma protein according to the increasing molecular weight on this side and plasma protein concentration on this side, then this is albumin. What is this? This is albumin, and these are globulins. Am I clear? Now, out of this is alpha globulin, these are beta globulins, these are gamma globulins. It means primarily plasma proteins are albumin and globulins can be separated. Globulins are globular protein, can be separated, right, according to their molecular weight into alpha globulins, beta globulins and gamma globulins. Is that right? Am I clear? Albumin's molecular weight is about approximately 70 kilodalton, but globulin's molecular weight is from 90 to 120 kilodalton. Right? Now, within this shade, where exactly antibodies are present? That is the question. Now, I think you should be intelligent enough to guess. In this band, this is albumin, alpha globulin, beta globulin, gamma globulin, and exactly where the plasma proteins are segregated. Israel, Dr. Safa. Of course, they are not alpha and beta, they are in gamma. Actually, if you study these plasma proteins, these are antibodies. If you study in this band, this is antibody. So, where are the plasma protein in your body? Plasma proteins are of course in the plasma, but within the plasma protein, where are the antibodies? Antibodies are part of the gamma globulins. Actually, that is why antibodies are also given another name. What is that name? Gamma globulins. Simple as that. Am I clear? So, there are three names. The antibodies, they are immunoglobulins, these are gamma globulins. Now, these are three same name for the same type of molecule. Is that right? That gamma globulins mean those globular proteins which are part of the gamma fragment, right, or gamma band on electrophoresis of plasma protein. Immunoglobulins mean what? Those globulin proteins which have immune function. They are also antibodies. And what are antibodies? Antibodies are those protein molecules which are secreted by well differentiated B cells called plasma cells, and antibodies specifically, this is very important, antibody molecules specifically react with the antigens which stimulated the formation of those antibodies. So, what are antibodies? Antibodies are protein molecules secreted by the plasma cells which react specifically with those antigens which elicited their or stimulated their formation or stimulated their synthesis. Am I clear? No problem up to this? Now, if I give you a statement, I say that antibodies are those globular proteins, right, which are produced by the plasma cells, and they react with specifically with the antigens which stimulated their formation. Plus, I can say antibodies are also called immunoglobulins. Because why we call the antibodies immunoglobulins? Because antibody molecules are just proteins which are part of the globulin components of the plasma protein. And which component of the globulins? Gamma globulins. Is that right? So next time, if someone asks you that in your blood where the antibodies are present, you say remove the cells, left is the plasma. Out of the plasma, separate the proteins, run the proteins electrophoretically and find the gamma band. And all your antibodies will be present in gamma band. Am I clear? There is no problem up to this. Now we come to really basic structure of antibody. As I told you, these are basically proteins with little carbohydrate component, and when proteins have a little carbohydrate component, we call them glycoproteins. When proteins have a little carbohydrate component, what we call them? Glycoproteins. To be more specific, antibodies are glycoproteins. Antibodies are glycoproteins. Now let's come to the basic structure of a typical antibody. Yeah. Antibody is basically a tetrapeptide. Any antibody is basically a, number one condition is, it is a tetrapeptide. When I say that antibody is a tetrapeptide, it means how many peptide chains it has? 4. It is tetrapeptide consisting of 2 heavy chains and 2 light chains. A typical antibody molecule is a tetrapeptide with 2 heavy chains and 2 light chains held together by, held together by disulfide bond. Disulfide bonds. So again, what is antibody? Antibody is a tetrapeptide. Antibody is of course glycoprotein molecule, right? It is a tetrapeptide consisting of 2 heavy chains and 2 light peptide chains. All of them are held together by disulfide bond. Am I clear? And it is usually a Y-shaped molecule. Let me make you a typical antibody molecule. Antibody molecule should have 4 peptides. Okay, I will make 2 heavy peptides here. Why we call this peptide heavy peptide or heavy chain? Right, this is a heavy chain and there must be another, yes please, another heavy chain. So antibody has 2 heavy chains. With that, it has 2, light chains. It has 2 light chains, and there are 2 heavy chains and there are 2 light chains, and they are held together by, yes please, disulfide bonds. These are the disulfide bonds which are holding them together, and of course they should be held together by disulfide bonds. Now let's go back to the definition. Antibody is of course an immunoglobulin which is secreted by plasma protein present in the plasma gamma globulin component, right? Antibodies are basically tetrapeptides. The 4 peptides having 2 heavy chains and 2, these are 2 heavy chains and 2 light chains held together by disulfide bonds. Am I clear? This is a typical, you can say, shape of the antibody. Now, antibodies have the specific function of the antibodies. They should bind with the antigen in a specific manner, right? Now, in which part of the antibody antigen binds? Yes, you will tell me. You could just come here and put your finger in which part of the molecule antigen will bind exactly. Yes, just tell it. Hurry up. I don't know. Top means how many points are there where antigen can bind? You don't know. Just come here. You know, be brave. Yes. Acha, antigen will bind here. Okay.
Like in this area?
No, I give you here. This is antigen, right? Very sad because it's going to bind with antigen. You draw it there exactly where it will bind. Okay, so it right over there. You were near the point, but honestly, it should be laughing here. You know why?
Why?
Because from here it can escape away.
What about
Your, no. It should be in between them like here, and then it will, you know, both chains will cooperate to catch it. You see? Antigens are not here, they will run away. Is that right? Actually, a part of a light chain and part of a heavy chain together grab the antigen. This is very important to know. This is one antibody molecule having two heavy chains, two light chains, right? And one antibody molecule, which I am showing here, can bind with two antigens or two epitopes of the antigen. Is that right? Now, let me tell you. Actually, the antigens which are the antibodies which are directed against different antigens must be different in this area. Let me tell you what I am telling. For example, here is one antigen like this, and other antigen is like this. Another antigen is do you think these antigens are same or different from each other? They are different from each other. And antibodies will bind with, do you think one antibody can bind with all three of them? No. Antibodies are very specific. The antibody which will bind with this antigen will not bind with the other ones. Is that right? Every antibody is very, very specific for a given antigen, a closely structural given antigen. Now, if I say that antibody which is going to react with this, it cannot react with this. An antibody which reacts with this antigen cannot react with the other two. It means this part of the molecule, this part of the antibody molecule should be different in different antibodies for different antigens. Are you understanding? For example, let me tell you that I will make a simple diagram that let's suppose this is like, now they can catch this antigen. Is that right? In the same way, now it will more specifically fit. Suppose this is antigen number 1, this is antigen number 2, this is antigen number 3. Is that right? Am I clear? Now, opposite to that, if there is another antibody and which will fit in, which antibody is directed for that antibody will have some difference. Now, do you think antigen number 1 can bind here? No, it is designed for antigen number 2. Antigen number 2 can better fit over here. Is that right? So what we see that this part of the heavy chain and the light chain is varying from antibody to antibody when antibodies are directed against for different antigens. So we say that let's suppose if we have 3 antibody molecules, one is for this antigen, other antibody molecule is for this antigen, and other antibody molecule is for this antigen, then what will happen? All those antibody molecules which are directed against different antigens should have this area with different configuration. It means this area varies according to the specificity for antigen reaction. That is why this part of the light chain, this is the light chain, this part of the light chain is variable light chain. And this part of the heavy chain also vary. So this is variable heavy chain. This component, this part of course, this is a peptide chain. So it should have here amino end and here it should have carboxyl end. The same way heavy chain should have one amino end, another JS carboxyl end. You understand this then? Every peptide chain has on one side amino end, another carboxyl end. So what really happens that amino end of the light chain and amino end of the heavy chain, they are having a domain. Domain means a region of amino acids sequence within a peptide chain. This domain is variable. So this is variable domain of the light chain, this is variable domain of the heavy chain. Is that right? But this component, this will not vary. It is constant. It is constant. We call it constant of the light chain and constant of the, yes please, heavy chain. This is variable of the heavy chain and this is constant of the heavy chain. And if you come to the light chains, this will be what? Variable of the light chain and this will be constant of the light chain. Is that right? No problem up to this? Let me make it more clear. Let's suppose there is star antigen, there is spherical antigen and there is potato antigen. The star antigen, ball antigen, B-A-L-L ball antigen and potato antigen. Now antibodies reacting against them will be same or different? Different antibodies. Let's suppose I show the antibody for this. These are the heavy chains, all of them. I am showing in a very simple way, you can understand. Now these are three antibody molecules, but antibody molecule which will react with the star, let me show star here, which is going to react with the star. This part of the light chain will be variable and this part of the heavy chain will be variable. Of course here if on one side it is binding with the star, on other side it will also bind with the star. So the same variable which is here on the heavy chain, here also same variable will be there and variable will be there on the light chain also. Now, if this molecule is going to be this ball antigen, then what will happen? Now, here is the, let's suppose I put the ball. Do you think the variable regions can be same as star? No. So they will vary from in this area. You understand it? Now we come to this. For that antigen, suppose there is another antibody which specifically react with that. Of course, it will have two heavy chains and just place two light chains, right? Now this antigen is like this. Now you understand it? That basically a particular antibody will react only with the specific antigen. Every antibody does not react with every antigen. And why a specific antibody molecule react only with the specific antigen? Because antigen binding area of the antibody has variable regions. And these variable regions, right, vary from one group of antibodies to the next group of antibodies to the next group of the antibodies as antibody group change their specificity for the antigens, right? Now come back. So this was variable domain of the light chain and this is the constant domain of the light chain. Then this is a variable domain of the heavy chain, this is the constant domain of the heavy chain. Heavy chain has many constant domain. Constant domain number 1, constant domain number 2, constant domain number 3, right? 1, constant domain number 1, constant domain number of the heavy chain number 2 or 3. And later on we'll find that most of the antibody classes, how many antibody classes are there? There are 5 basic classes of the antibodies, right? The 5 classes of antibodies are called, can you tell me? Immunoglobulin G, A, immunoglobulin A class, immunoglobulin M group class, immunoglobulin E and immunoglobulin D. We call it gamed. All the immunoglobulins can be primarily divided into 5 classes. I will tell you later how we divide them, on what base immunoglobulins can be divided into 5 classes. But antibodies can be divided into 5 classes. This is immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin Ig. Now you understand why we call it immunoglobulin. These are the globular proteins from the globulin component of the plasma proteins having immune function and belonging to the class of G or class of A, right, or class of M, E or D. Out of this, Me, very important, you know. Me has longer heavy chain. What does it mean? That basically immunoglobulin M and immunoglobulin E has heavy chains with constant number 1, constant number 2, constant number 3, constant number 4. Right? So, constant of the heavy chains in immunoglobulin class M and class E is how many? 1, 2, 3 and 4. But immunoglobulin A, G and D, their heavy chains are having only how many constant domain? Constant domain number 1, 2 and 3. But IgM and IgG have how many? 1 additional, that is 4. Is that right? Now let's review it. Antibody molecule has how many peptides? A typical antibody molecule has how many peptides? 4. 2 are heavy chains and 2 are light chains. Why light chains are called light chain? Because the molecular weight is less. Usually light chains molecular weight is about 25,000 and molecular weight of the heavy chain is 50,000 to 70,000 depending upon, there are 3 constant or 4 constant region, right? So there are 4 peptide held together by the disulfide bond, 2 light chains, 2 heavy chains in a typical antibody molecule. Is that right? Now, on the amino ends of the heavy chain and amino end of the light chain, there are which areas? Variable areas. There is variable area of the light chain, there is variable area of the heavy chains. In between these 2 variable area, what is fitting there? Antigen fits. Is that right? So antibody binds with the antigen through the heavy chain as well as through the light chains through its variable domains. And then there is only 1 constant domain of the light chains, but heavy chains have how many? 3 or 4 constant domains. Any question up to this? There is no question, no problem up to this. Now, another thing, that some doctors, they were doing some experiment with the antibody molecules, and what did they do? Let me tell you, suppose I draw here a typical antibody molecule. I hope you understand it, isn't it? And what did they do? That put these molecules in a flask, right? Put the antibody molecules in flask. But in the flask, in this container, after putting the antibody molecules, I think I will make the light chains color different so that you can appreciate the diagram better. These are the antibody molecules which we have put into this container, and of course they are held together by, yes please, disulfide bonds. Now what the researchers did, they added a very naughty type of enzyme. What is this enzyme? This is called papain. They added a papain to this, and they were surprised that papain could break down this molecule at a special points. Okay, I will keep my color scheme same. Right? They found that papain could cut this molecule at this point. It digested the molecule of antibody. Antibody is a protein that was digested by this papain. And papain is an enzyme which attacked this region. This is called hinge region. What is this? H-I-N-G, hinge region. Attack the hinge region and digested from this point and this point. Now antibody break down into how many components? How many fragments? Yes, question goes to Mr. Yes. How many antibodies broken into how many components? The 3 components. Look at it. One component is this, another component is this, and another fragment is this. So antibody break down into 3 fragments. So what did they do? They put antibody molecules into container and add the papain to that, and papain digested every antibody molecule into how many fragments? 3 fragments. And these fragments which were made, out of them this fragment has, right, amino end of the heavy chain and complete light chain, right, held together. Another amino end of the heavy chain with complete light chain. And this portion has remaining heavy chains held together by disulfide bond. Is that right? Now, then after that, they added the antigen to this. What did they add? Let's suppose this is the antigen. For that particular antibody, they added the antigen to this solution. And they came to know that these fragments, antigen bind with these fragments. Right? But do you think antigen will bind at this point? Or will the antigen bind here? No. Will antigen bind here? Will antigen? Yeah. Will antigen bind here? Will antigen bind here? Will it bind here? But it will bind here. So in this way, the researcher found that when antibodies are put into a container and treated by the papain, antibodies will break down into fragments. Some of the fragments will be binding with the antigen and other fragments will not bind with the antigen. These fragments which do not bind with the antigen, they found they will go to the bottom of the flask. These fragments will go to the bottom of the flask, and there they will crystallize. They will change into crystals. Due to that reason, doctor said when an antibody is treated by the papain, when an antibody molecule is, again let's draw it here and here, this is disulfide bond. Here is your light chains, isn't it? So they found when they treat with the papain, antibody will break down at this point. Is that right? It divided into 3 fragments. Out of these 3 fragments, 2 fragments were able to bind with the antigens, and other fragment was unable to bind with the antigen. And this fragment went down and it crystallized. So they called this fragment of the antibody fragment which crystallizes. FC portion of the antibody. In the books you very frequently read FC portion of the antibody or FC fragment of the antibody. C originally was put for the crystallization, that this component of the antibody will crystallize once antibodies are treated with the papain. And this fragment, this will binding with the fragment which was antigen binding. We call it fragment antigen binding. So we call it FAB. So basically a classical molecule of antibody consists of 4 peptides having 2 heavy chains, 2 light chains held together by disulfide bonds and having 2 FAB fragment and 1 FC fragment, right, or portion. Am I clear? Now, actually this FC stands for crystallization, but it also should remind you that with this end there is what? Carboxyl end of the heavy chains, because amino end was here for the heavy chains. So C for crystalline, C for carboxyl end, C for complement activation. Some of the antibodies activate the complement. This is the portion of the antibody which activate the complement. This is the portion of the antibody which activate the complement. It means in this domain, more specifically, if you remember, there was for heavy chain, how many domains were there? Heavy chain, variable, constant, heavy chain 1, constant, heavy chain 2, and constant heavy chain 3, or some cases 4. Actually this is the constant heavy chain 2 which has the capability to activate the complements. So complement activating part of the antibody is part of the FC fragment. So FC fragment should remind you that that this portion crystallizes, this portion is having carboxyl end of the heavy chain, and if this antibody is going to activate the complement, then FC portion will activate the complement. And you will know later that IgG class of the antibody crosses placenta. IgG class of the antibody from the mother through the placenta goes to the fetus. So actually with the placental cell, this portion of the antibody bind and help the antibody to cross through the placenta. So it should remind you also about cross through, yes, placenta. And antibodies bind with specific type of cells. For example, later on you will learn IgE specially bind with catches the mast cell. Is that right? So again, IgG class of antibodies will catch the mast cell or bind the mast cell with this portion. So catching cells. Am I clear? No problem. So FC portion stand for what? Originally standard for this fragment could crystallize, but this should also remind FC stand for carboxyl end. FC stand for fragment which activate complements, fragment which help the antibody to cross the placenta, and fragment of the antibody which help it to bind with the cells. Any question up to this? And FAB portion, it's enough to say that this is a fragment which is antigen binding fragment. Am I clear? No problem up to this. Okay. Then, so we can say that one antibody has what thing? Antibody plus papain, yes, goes to what thing? Antibody will go into what? Antibody treated by papain will go into 2 FAB fragments plus 1 FC fragment. Is that right? Now we do another treatment. Take an antibody molecule, okay, let's make some seductive color scheme. Is that right? This is a typical antibody molecule, of course, consisting of 2 heavy chains and 2 light chains. Is that right? Now, rather than papain, this time we treat it by pepsin. You have heard of pepsin? What is pepsin? It is also proteolytic enzyme, not a name of a girl, pepsin, right? When antibody is treated by the pepsin, at which point pepsin will digest the antibody? It produces multiple digestions at this point. So actually now, when antibody is treated by the pepsin, then FC portion is digested at multiple fragments, but both FAB portion are still kept together. The important point is that pepsin attack the antibody molecule not at the hinge region, rather distal away from the hinge region towards the carboxyl end of the heavy chains. Is that right? Due to this reason, both FAB portion are kept together. Am I right? Now you will help me rapidly. If I say this is one antibody molecule and this is another antibody molecule, let me take your test. Enzyme attack here and divide the antibody molecule into how many fragments? 3. Which enzyme is this? Yes? Pepin. And if I say that enzyme attack here and both FAB portion are still held together, what is this? Pepsin. Am I clear? No problem up to this? For sure? Okay. So now we have learned something about the basic structure of the antibody. Another thing, then when antibody is reacting with the antigen, when antigen and antibody bind together, they are held together by what type of forces and what type of bond? You know, when they come together, they make a bond, isn't it? So what type of bond is present between the antibody and antigen? Yeah, I'm about to be impressed by you. Okay, let me make our original molecule again. I hope you remember what are these chains I'm drawing? Heavy chains. And what are these chains? Yes, light chains. And rapidly tell me, what are these bonds I'm making? Disulfide bonds. That's good. And what is this fragment? Variable of the heavy chain. And this is also variable of the heavy chain. And what should be here? This area? Variable of the light chain. And what should be here? Variable of the light chains. Then let's suppose I'm going to make the antigen. If antigen is like this, right? Now this antigen is if going to fit here, then what will happen? What was this area? Constant or variable? Yes? Variable. Now when this will fit into this, I think I have to make this a little lengthy so that it should look more proportionate with the yes, now this is caught over here and right? So we say antigen is caught. It's caught in between the variable region of heavy chain and variable region of the light chain. This is what you already know. Now I know a little more. Actually, within the variable region, there are very special areas which make the fitting of variable region more better with the antigen. And these areas are given an additional name. You know, this is making a better fit, isn't it? These areas are called hypervariable regions. What are these called? These three are called hypervariable regions. So we say within the variable region of the light chain and within the variable region of the heavy chain, there are small amino acid groups, right, which are called hypervariable regions, which make the binding between the you can say antibody and antigen as precise as possible. Very, very precise. Is that right? Now I will draw the same thing here and you will tell me what's going on here. Yes, now you will tell me that this is variable of the heavy chain and this is variable of the light chain, and antigen is caught over right. But within the variable of the light chain, what is this region? Hyper variable regions. Is that clear? One thing which is very important to know, that in a given antibody molecule, the antigen which bind on the same side, one side, that is the same antigen is going to bind on the other side as well, right? So both sides have the same antigen specificity. Am I clear? No problem up to this? Right. Then I was talking about with which forces they are held together, or which type of bonds are in between them. What type of bonds are in between them? The forces are the forces with which antibody and antigen are interacting. Those forces are very simple forces. These forces are, yes, have you heard of electrostatic forces? Electrostatic forces, let me tell you how they are coming. For example, amino acid which is present over here is positively charged and amino acid here is negatively charged. They are attracted to each other. You see they are held together by electrostatic forces or by the van der Waals forces. You know van der Waals forces? Van der Waals forces. So there are electrostatic forces or there are van der Waals forces. This wall is not that wall on which you sit and fall down, right? This is V. So electrostatic forces and van der Waals forces keep the antigen and antibodies together when they are specific for each other. And what type of bonds are formed in between? Is this ionic bonds or are they covalent bonds, right? Or they are like simple hydrogen bonds and hydrophobic bonds. Yes. Yeah, Dr. Safa is trying to tell. You know, the bonding between the antibody and antigen is, one way to tell is that is it strong bonds or weak bonds? Okay, only tell me this thing. They're weak bonds, right? If you bring, you know, let's suppose if there are, you bring lot of antibodies, right? Some of the antigens may dissociate and go to the other antibody if all the antibodies have same antigen specificity. So these bonds are not permanent bonds, they are somewhat reversible bonds or they are weak bonds. So we can say that these bonds are not ionic bonds, these are not covalent bonds, these are simply, yes, hydrogen bonds and hydrophobic bonds. So the forces which are keeping the specific antigen antibody molecule together are van der Waals forces and electrostatic forces, and making very weak bonds like hydrogen bonds and hydrophobic bonds. Am I clear to all of you? Is there any special question here? There is no. Okay, let's have a break. Now some more basic concept about a typical antibody molecule. Right, so we were talking about that antibody molecule, the tetrapeptide. Now we have to develop a relationship between tetrapeptide We should be clear about these terms. And heavy chains, light chains, fragment FAB, fragment C, and then we should develop a concept what is monomer, monomer molecule, dimeric molecule, dimeric antibody molecules, pentameric. I will teach you this. Pentameric molecules. What is the concept of affinity and avidity? Right? And what is the concept of valency? You already know about the tetrapeptide. We said that every antibody molecule is basically a tetrapeptide made of 2 heavy chains and 2 light chains, right? And a typical molecule has 2 FAB fragments if it is treated by papain and 1 FC fragment. These terms I will explain now. We go back to our typical molecule. That these were heavy chains and these are light chains. All of them are held together by disulfide bond. Now, actually, this whole tetrapeptide is considered one monomer. This is considered one monomer, and in simple diagrams, it is shown like this. Actually, these part of the Y are representing FAB fragment, and this stem of the Y is representing FC fragment. That is why very commonly you find antibody molecule like this, right, made in the books. So basically when you see an antibody molecule like this, it is fundamentally a tetrapeptide, but when these 4 peptides are held together by disulfide bond, we say this antibody molecule is one monomer. It is monomer. Is that right? Now sometimes it happens that more than one antibody molecules are held together, they are held together. This is one monomer, right? And this is, suppose, another monomer. And if these monomers are held together by another peptide chain, this peptide chain is holding them together. This peptide chain is joining them. So we call it joining component or J component. What we call it? J component. If more than one antibody monomers are held together by J chain or J component, right, or J chain, joining chain, we say now we are having multimeric molecule. Now we are having multimeric molecule. And this multimeric molecule has 2 monomers, so we call it, this multimer is dimer. This multimer is dimer. Sometimes what happens that with the joining component there are one, 2, 3, 4 and 5. Now in this case, this J component is holding how many chains together? It is holding 5 chains together. So we call it pentamer. This is this type of you can say grouping of the antibody molecules, right, in which 5 monomers are together and held together by the J chain. This is called pentamer. So when we are talking about these terms, when I say that there is this antibody is monomeric, it means this antibody exists in this form. If I say there is another antibody which exists in dimeric form, it means it exists in this form. And if I say that a particular antibody group is pentameric, it means it is existing in this form. Now we have to develop the concept of valency also. Valency means that one group of antibodies can bind with how many antigens. For example, this can bind with how many antigens? Only with 2. So we say this monomeric molecule is having 2 valencies. So we say this monomeric molecule is divalent. And this monomeric molecule is tetravalent. valent. And this monomeric molecule is, yes, it can bind how many antigens? 10. So it should be decavalent. You understand it? So now you should know that when we are talking about antibody molecules, these special terms are meaning exactly what concept. When we say an antibody molecule is tetrapeptide, it means a typical antibody molecule has 2 heavy chains and 2 light chains. When I say that antibodies of different classes may exist as monomer or dimers or pentamers, it means we are talking about that. If I say that, for example, immunoglobulin IgG, it exists in monomeric form, it means there is no J chain holding multiple molecules together. If I say that immunoglobulin A group in the body secretions are dimeric, it means they are held together by J chain, 2 monomers are held together by. Or if I say that immunoglobulin class of IgM in the serum is in pentameric form, it means it is holding how many? 5. But if I say that, let's suppose this molecule belongs to immunoglobulin G class, so we say this is monomer with divalency. And if it is IgA, this is dimer with tetravalency. If it is IgM molecule in the blood or plasma or serum, then it is pentameric molecule with decavalency. Decavalency. Am I clear? Any confusion up to this? There is no confusion? Okay. So this concept is clear that valency refer to the group of antibodies binding to how many groups of antigens. Is that right? Or one antibody can bind with 2 antigens. It is divalent. But if it is dimer, it will have tetravalency. If it is pentamer, it will have decavalency. No problem. Now we have to come across another term, concept of affinity and avidity. Concept of affinity and avidity. Let me tell you that what is the meaning of affinity between the antigen and antibody. Right? Affinity. Let's suppose this is one antibody molecule. This is another antibody molecule. Now both of them, okay, first I will make the antigen here. This is the antigen and here this is the antigen. Now this antibody molecule is having these variable, what are these? Variable regions which are binding with antigen. Now we make another antibody molecule and this is what? Antigens. These are its which portion? Let's suppose this is antibody molecule number 1, this is antibody molecule number 2. What do you think? Antibody molecule number 1 is holding the antigen more tightly or antibody molecule number 2 is holding the antigen more tightly? 2. Actually, these interactions between the variable and hypervariable region of antibodies and between the antigens, these interactions, they determine how tightly antibody will hold the antigen, right? This tightness or strength, the strength or the power with which one antigen binding site binds the antigen, this strength is called affinity. So molecule number 1 will have less affinity for the antigen and molecule number 2 will have more affinity for the antigen. Is that right? For example, there is a beautiful girl here and her sister is holding her hand. Affinity will be more or less as compared to her boyfriend, a very new boyfriend holding her hand. Where affinity will be more? New boyfriend, not old one. Alright? So what I'm saying that affinity means that if antigen antibody are interacting, how tightly they are holding each other together, or how what is the strength of the power with which one antigen binding site is interacting with the antigen, or as Shan is saying, exactly what is the binding force, between the antibody molecule and antigen. Am I clear? This concept is called affinity. But avidity is a slightly different concept. Affinity refer to the binding force between antigen and antibody at one point. Avidity mean a different thing, let me tell you. Let's suppose that there is a very big antigen. It's a very big antigen with multiple points with which antibodies can, yes, it is okay, I will make it a simpler diagram so that you don't become afraid of this. Okay. This is a very big antigen with small epitopes. Epitopes are the points exactly with which the antigen binds. Now look, if a monomeric antibody come, this will react here and here only at 2 points. And if polymeric or multimeric antibody come, the J chain. Do you think monomer has only 2 points to bind, but dimer has how many points to bind? 4. And if pentamer is there, how many points of binding? Especially in large antigens, 10. So total force with which I will repeat it again. This is monomer, this is dimer. Monomer is actually divalent and dimer is actually tetravalent, right? Affinity depend on, when we talk about affinity, we are just talking about that one antigen point and one antibody point interaction. But when we talk about avidity, we are actually looking at all the affinities in a group together. This binding force plus this binding force plus this binding force plus this binding force. Affinity at 1, affinity at 2, affinity at 3 and affinity at 4. All affinities together are called avidity. What are they called? Avidity. Is that right? So, again I will repeat. Avidity means that at how many points antigen antibody are interacting. As IgM molecules, they are pentameric, so their avidity is more. But IgG molecules are only monomer, so they are having only divalent, so their avidity is less. Am I clear or not? No problem up to this? Okay. Now we develop some concept about the different classes of antibodies. So antibodies have, all of you know that basically antibodies are having 5 main classes. Now according to which concept, what is the reason to divide your antibodies into 5 classes primarily? You have any idea? Why antibodies are primarily divided into 5 classes? There must be a reason. Yeah? No, that is not the real reason. Why antibodies are, for example, look, you bring from my blood, again, from my blood you bring all the plasma protein out and run the plasma proteins on the electrophoretic strip, and from the gamma band you bring all my antibodies out. All the antibodies which you bring out of my gamma component of the plasma proteins, they can be basically classified into 5 groups. On what base they are classified? Yes? Yeah, that's good, very good. Actually, primarily 5 types of heavy chains can be produced and 2 types of the light chains can be produced. The plasma cell can produce primarily how many types of heavy chain? 5 types of heavy chains and 2 types of light chains. Now, if you if you put all my antibodies out of my bodies, you bring all my antibodies out and classify them according to the heavy chains, how many groups will be found? 5 groups. Let's suppose this is one group of heavy chain, this is another type of, okay, I will make it a diagram and then I will explain that why I am making it like this. These are just, I am drawing the heavy chains. How many types of mainly heavy chains our B cells can produce? Mainly 5 types, even though there are subgroups also. We'll talk about that. 1, 2, 3, this is the 4th and 5th group. Right, so these are the types of heavy chains which can be produced, and the light chains which can be produced are only 2, the kappa and lambda. Now, antibodies are classified according to the heavy chains into how many classes? 5 classes, because mainly 5 types of heavy chains can be produced. So if you put all my antibodies in the table, and if you can look at their FC portions are made of heavy chains. FC portions are made of heavy chains. How many different types of heavy chains you will find? Primarily 5. Is that right? So we'll say they are basically 5 types of heavy chains and 5 classes of immunoglobulins primarily. Is that right? But light chains are only of 2 types, kappa and lambda. Heavy chains are like, suppose this heavy chain is called gamma chain, this heavy chain is called alpha chain, suppose this heavy chain is called mu chain, this heavy chain is called delta chain, and suppose this heavy chain is called epsilon chain. This heavy chain is called epsilon chain. So basically antibodies can be classified depending upon the type of heavy chains into 5 classes. But if you look at their light chains, there are only 2 types of light chains. There are kappa light chains, suppose this is kappa light chain, and another is lambda light chains. Actually, if you remove all the antibodies with the gamma heavy chains, some of them will be having lambda light chains, of course held together by disulfide bond, and some of them will be having lambda heavy chains, light chains, is that right? So actually kappa and lambda light chains are present in every group. So we can say some of the molecules in every group will be having, what is this? Kappa light chains, and some of the molecules will be having lambda light chains. Is that right? And of course you are so intelligent, all of them are held together by disulfide bonds. There is no need to make it, you understand it. It means if you have these 5 group of antibodies or 5 classes of antibodies, why the antibodies in different classes are different? Because they have different heavy chains. And why antibodies in different classes have some similarity? Because they have same light chains. Again listen, we say that antibodies are basically having 5 classes. Why we divide the antibodies into 5 classes? That classification is depending upon the types of heavy chains. Is that right? So in one group, in any one group, suppose we talk about this group, if we take antibodies of only this group, all the heavy chains will be same. Is that right? But some light chains will be kappa and some light chains will be lambda. So when you study in comparison group to group, why groups are different from each other? Because they are having different heavy chains, but they do have some physico-chemical characteristics which are similar, and they are due to light chains. Am I clear? No problem up to this. Now, those antibodies which are, those immunoglobulins which have gamma heavy chain, they are called IgG. Those immunoglobulins which have alpha heavy chain, they are called IgA. And those immunoglobulins which have gamma heavy chain, they are called IgG. immunoglobulin G. Sorry, this is mu. So they are called IgM. And those immunoglobulin molecules which are delta chain, delta heavy chain, they are called IgD. And those immunoglobulins which are having epsilon heavy chain, they are called IgE. That is why we say there are 5 classes of the immunoglobulins, because there are 5 different types of primarily heavy chains. Then later on you will learn that there are 2 subclasses of IgA, that is IgA1 and IgA2. And in the same way IgG can be divided into 4 types: immunoglobulin G1, immunoglobulin G2, immunoglobulin G3 and immunoglobulin G4. Is that right? Any problem up to this? So these are the subclasses. IgG has 4 subclasses, IgA has 2 subclasses. No problem up to this? Now let's go into detail of every class one by one. We'll start studying now in detail every subgroup. First of all, we'll discuss about IgM. Important points related with immunoglobulin IgM. Immunoglobulin IgM. Of course, you will tell me that IgM has what type of heavy chain? Mu type of heavy chain. Now another question which I want to make it clear. Before I really go into detail of classes, let's suppose you have a bacteria, and on this bacteria, one bacteria has only one type of antigen, or one bacteria may have many types of antigens. One bacteria may have one type of antigen, or one bacteria may have many different types of antigens. Yeah. One bacteria, suppose this is one bacteria, it may have star antigen, it may have ball antigen, it may have triangular antigen, it may have square antigen. Actually, one microbe may have different types of antigens. That is why we say microbes are antigenically mosaic. Right? Every microbe has multiple type of antigens. Now, of course, the antibody which is made against a microbe, that will be one type of antibodies or multiple type of antibodies against. For example, this bacteria enter into your blood or into your body. Suppose this is E. coli. Okay, this is spherical, so it cannot be E. coli. It is Staphylococcus aureus. It enters into your body, and you build an immune response. Right? This immune response will have antibody directed against only one antigen, or antibodies will be produced which will be against multiple antigens. So there will be anti-star antibody, there will be anti-gola antibody, or anti-ball antibody, maybe anti-triangular antibody, anti-square antibodies. What I am saying, that against a microbe which has multiple antigens, it is quite possible that you will build immune response against multiple antigens. This type of immune response is called polyclonal immune response. One clone of plasma cell or B cell will produce anti-star antibodies. Another clone of B cell or group of B cell will produce anti-ball antibodies. Another group of B cells will produce anti-triangular antibodies or anti-square antibodies. Am I clear? So usually against an organism, the antibody response is polyclonal. Right? That microbes are antigenically mosaic, and immune response from a healthy person, immunocompetent person, is polyclonal immune response. Now another thing, let's suppose we talk only about anti-star antibody. Let's suppose this star antigen come into your body and we are making anti-star antibody. This anti-star antibody will belong to which class? It will belong to IgG or IgM or IgA, or which class? It can belong to any group. Against one antigen, we may make IgM in the beginning, and later on we can make IgG or even IgA. This is very important concept, extremely important. Against the same antigen, at different times or simultaneously, you may be producing antibodies of different classes. Now let me make this concept more clear to you. Let's suppose this is star antigen, Now, these are suppose heavy chains are suppose mu heavy chain. Oh, okay, these are gamma heavy chains. If these are gamma heavy chains, then can you tell me which is the antibody which is reacting with the star? Which class of the antibody is reacting with the star? If it is gamma, IgG. This is IgG molecule because it is having a heavy chain belonging to gamma. But the interesting thing is that now heavy chain has been changed, but specificity for the antigen has not changed. Is that right? And now let's suppose it is mu. If this heavy chain is mu, then what is this antibody? Class IgM. Let's suppose that this heavy chain is, yes, alpha. If these are alpha heavy chains, then what is this? IgA. You see, against the same antigen, you can produce IgM, IgG, IgA. Are you understanding me? Right? So against one antigen, you can produce antibodies with different classes. Usually the first class of antibody which is produced is IgM, and later on you produce IgG against the microbes. When microbe first time enter into your body, in the antibodies you use which heavy chain? Mu. Usually when antigen is entering in your body first time, when plasma cells are making antibody on first exposure, they use which heavy chain? Mu. Because they are using the mu heavy chain, so antibody class which is initially synthesized against antibody is IgM. But later on class switching occur. The same plasma cell may start producing IgG. Now you may be thinking how it is possible. Have you ever thought of it? How this class switching occur? How one class of antibody switch to another? Let me explain very briefly. Later on we'll have a full lecture on class switching and genetic. Suppose this is a plasma cell, right? This is the nucleus of the plasma cell. Let's suppose here are the genes for the heavy chain. You know, in the heavy chain, this is a portion, this will make which? Mu. When this part of the genetic material expresses itself, you make which chain? Which heavy chain you make? Mu. And when this start expressing itself, you make which heavy chain? Delta. And when next gene, let's suppose this gene start expressing itself, what is this? Gamma. So this is the gene for mu, this is the gene for delta, this is the gene for gamma, and this is the gene for alpha, and this is the gene for epsilon. Is that right? Now these are the genes which are present in every plasma cell to synthesize a special type of what? Heavy chain. Is that right? Now let's suppose this is a special part of gene, and this part of the gene is concerned with the heavy chain variable, and this is heavy chain constant for the mu, this is the heavy chain constant for the delta, this is the heavy chain constant for the gamma, this gene is for heavy chain constant for alpha, and this gene is for heavy chain constant for epsilon. Now you listen very carefully. When first time antigen come, right, this part of the DNA will combine with the mu. So antibody which will be formed, that will be, suppose this part of the DNA makes the variable portion of constant which will react with star. Now this is suppose make the peptide in the heavy chain which will react with, bind with the star antigen. Is that right? This is the genetic component of the B cell or plasma cell, that when it translates into a peptide fragment, that piece of the peptide will become the variable portion of the heavy chain which will react with a special type of antigen which is suppose star antigen. Now what really happens? Initially this fragment fuses with this and both of them come together and start making heavy chains. So in the beginning the heavy chains which are made in this cell, they will have this portion with this portion. And what is this? This will be actually, if I really put it like that, what is this? This is a heavy chain for the IgM. Is that right? So in the beginning, this cell will be making heavy chains for the IgM. And the antibodies which will be produced, of course, what will happen? This heavy chain with another mu heavy chain with two, let's suppose, kappa, and what will this become? Whole molecule will become IgM molecule and it will be secreted out. But if antigen again and again come and exposure become prolonged or repeated, this will be digested away and thrown out. It will change its strategy. And this portion will fuse with this one. Both of them are thrown out. Both of them are thrown out, loop out and destroyed. Now the combination will be made VH with gamma. Now the heavy chain which is made now, this is having this, this gene and this gene fragments will come together. And new combination will make the peptide chains, right, which will still bind with the star. So bind with the same antigen. But of course these are the light chains. But heavy chain has been changed. The constants of the heavy chain has been changed. And the class of the antibody has switched from IgM to IgG. You are understanding? So against the same antigen, plasma cells are initially producing IgM and later on they switch the class and start producing IgG. So against the same bacteria and against the same antigen, initially you may produce IgM and later on you may produce IgG or you may produce IgA. Am I clear? No problem into this? Is that right? So this is the concept of class switching. Because this portion of antibody is different from class to class, right? But this portion of antibody may be same for a given antigen, even classes may be different. Am I clear? And due to this reason, whenever you get recent infection in your blood against that infectious agent, IgM level will go up. But if infection is repeatedly there, then IgG levels will also go up. So if you are having some disease, right, and I checked your blood and against an antigen you have only IgM, it means you had recent infection. But against another disease, I checked your blood and I find IgG but I do not find IgM. It means there was some previous infection. Are you understanding? And if I find against a bacteria IgG and IgM, IgM means infection is going on. IgG means it's quite long time. You are understanding it? So it's prolonged infection. Again, let me tell you something. If this is my friend, this marker is my friend, against a bacteria it has only IgM. It means it got bacteria recently. This is my friend, against the same bacteria it has only IgG. Probably it has previous infections. This is my friend, against the same bacteria it has IgM and IgG both. It means he has infection from long time, that is why he has IgG and still it is present in his body and he is still making IgM. Am I clear?
Could it also mean that he had the infection before and then he recovered and he is getting a recurrence?
It is quite possible. It is quite possible. Someone who has against a specific antigen IgG and IgM, maybe IgG represents the past infection and IgM represents recent infection. Am I right? No problem up to this. Here, Now we go to details of the antibody classes. Now we'll go to the IgM molecules. First of all, we'll discuss IgM, class of the antibodies or immunoglobulins. The very important thing that IgM is normally expressed on the membranes of the B lymphocytes. This is the B lymphocytes, and on the surface of the B lymphocytes, IgM molecules are expressed. What is the purpose of expressing the monomers of IgM on the surface of B cells, B lymphocytes? Yes, I will be impressed if you can tell me. Okay, let me tell you before you tell me something new. So this is one B cell, this is another B cell, this is another B cell, right? Let us suppose this B cell is presenting IgM on its surface, and this IgM can bind only with the ball antigen, right? This B cell is presenting IgM on the surface, and this IgM can bind with the star antigen. This is another B cell, and this B cell has IgM molecules which could bind with square antigen. Actually, every B cell in your body expresses IgM molecules on their surface, and different IgM molecules on the surface of the different B cells bind with different antigens. Let's suppose these three B cells are there and there is no antigen, and these three B cells are there in your body. If star antigen come, this will be activated. If ball antigen come, only this will be activated. If square antigen come, only that will be activated. So actually, there are millions and millions of the B cells having special type of IgM on their surface, and these IgMs are going to act, these IgM surface IgM molecules are going to act as a receptor for the receiving molecule, receptor for the antigen. That is why these surface IgM molecules are called BCRs, B cell receptors for the antigens. So next time someone say that B cells are having receptors for the antigens, most of these receptors for the antigens are actually IgM molecule. Some of these may be IgD molecule. So IgM mainly, and sometimes IgD also act as B cell receptors, right? So actually different B cells have a capability to produce different B cells have a capability to produce antibody for which are specific for different antigens. All B cells cannot make antibodies for same antigen. Is that right? So actually every B cell on its head is having a sample antibody, So actually antigen goes and bind with the relevant, and then that B cell will be activated and convert into plasma cell. Now if star antigen come, only this B cell will convert into plasma cell. But if square antigen come, do you think this will be activated? No, that will be activated. So right, so what really happens that antigens come and bind with only that B cell which is having such IgM or IgD molecule which can react with specific type of antigen. So we can say one function of the IgM molecule is that they act as antigen receptor on the membrane of B cell, and their IgM molecule is present as monomer. So these are acting as BCR. So this is one important action of IgM, right? But IgM is not only present on the surface of the B cells, which is called surface IgM or membrane IgM or B cell IgM. They are monomeric. But actually IgM molecules are actively secreted by plasma cells, and IgM molecules are present also in your blood. in your serum, and serum IgM is pentameric. They are not monomeric, right? Suppose this is your blood. This is your circulation. In your circulation, let's suppose this star is the J chain. Then how many IgM will be there? 1, 2, 3, 4 and 5. Right? So basically, IgM molecules are present in our serum or plasma as pentameric molecule which are joined together by the J chain. Is that right? Because they are large molecule, they are mostly present in the blood, they are not present in interstitial fluid, and they are not part of other body fluids. IgM is mostly distributed in plasma. Is that right? This is the another thing. That IgM, here it is pentameric, but it is decavalent. I told you IgM is the first antibody which is produced during the immune response to a new antigen. Right? Such immune response is called primary immune response. When first time you get antigen and you develop antibody, we say it is primary immune response. But if antigen is coming repeatedly, then you are developing more immune responses. After the first immune response, whatever later on immune responses come, they are called secondary immune responses. Let me tell you here that let's suppose you are vaccinating a person. You are giving vaccination. This is first vaccination, second vaccination dose, third dose. During the first dose, you will develop gradually IgM, but then IgM level will go down. But with every dose, it may go up. But during the first response, the IgG will be very less. In second response, IgG will become very high. Third response, it will become very high. Now listen. This, when antigen first time enter into your body, you develop an antibody response after some delay. After some delay. From here to here, there was no antibody. Gradually it started. This is called lag period. What is it called? Lag period. So when first time antigen come, there is a long lag period, and then antibodies will start appearing in the blood very slowly. Is that right? And they will go a very low level of peak, and then gradually they will come down. This immune response is called primary immune response. And in the primary immune response, main antibody is IgM, and IgG and other antibodies are very less. But if you repeat the, okay, I'll make it more clear. Let's suppose first dose, second dose, third dose of vaccination. First response is right. This is IgM. So you see, with every time antigen comes, there is a little bit IgM response. Is that right? And initially this is first response, but IgG response, this is very little in the beginning. Second time it is very rapidly going up, goes to a peak, stay for a long time, come down, but not so much. And then next dose, third dose, it goes very high and stay for very long high. So actually, we say with repeated antigen exposure, IgM is not boosted, but IgG is boosted. Is that right? So when you give me star antigen first time, I will have mostly anti-star IgM, but very little anti-star IgG. But after a few months, you repeat star antigen, then I will have still little IgM, but lot of anti-star IgG. And this time it will come very rapidly, it will go very high level, and it will come down and stay on the high level, right? This type of response is called secondary response, but this type of response is primary response. So on first exposure of the antigen, antibody level go up slowly, goes to a very low peak and gradually come down and persist at very low level. But on repeat exposure, antibody level goes very rapidly, very high, achieves very high level, stays there for longer time and come down slowly. Right? On every repeated exposure, this will behave that very briskly antibody level go up and stay very high for longer time. This first response is called primary response. The second response is called Secondary response, and this third response is called, yes, Dr. Safa? It is not called tertiary. First response is primary response. After that, every repeat exposure is secondary response. Characteristics of primary response are long lag period, very few antibodies formed, and it comes down and stay very low. This is primary response, and main antibody is IgM. But in secondary response, what happened? Yes. There is short lag period, very rapidly rising antibody level, very high peak is achieved, which stays for longer time and come down, but stays at higher level. This type of pattern is called secondary response. Secondary response has main antibody of IgG class. Why it happens so? Look, when first time some plasma cells started, they were mainly producing IgM. You know, bone marrow is producing the B cell. After 6 months, when second dose comes, some new B cells have come. They again produce IgM. Then after 1 year, again exposure occur. Then again some new come and IgM. But from where the IgG come? IgG come when these first IgM have done class switching. So next, these are called memory cell. So next time when antigen come, previous memory cell very rapidly bigger response. So these are the rapid response by the memory cell which were in the previous exposure IgM producing cells. Now they are producing IgG. When you come third response, then memory cell from here and memory cell from here, both of them respond. So every time when you are boosting, you are increasing the memory cell against the antigen. And these memory cells are having what? Capability to produce more and more IgG. That is why IgG keep on getting boosted with every response. But IgM does not get boosted. Am I clear? No? No problem up to this? Now we were talking about that, so we can say that IgM is surface IgM, which is acting as B cell receptor. Number 2, there is serum IgM, and about serum IgM what you should know? That it is pentameric, yes, pentameric. There are 5 monomers of IgM held together by J chain, and this is decavalent, because it can bind with how many antigens? 10. So it is having less avidity or very high avidity? It has very high avidity. Is that right? So because of another thing, because it is decavalent and it can react with multiple antigens. Now you imagine, let's suppose this is IgM against your antigens on RBCs. If this is IgM pentamer against the antigen on RBCs, it will react with one RBC or multiple RBCs. And one RBC may be reacting with multiple pentamers. So what is happening? So what really happens that, are you understanding what is happening? The network RBCs which were originally separate from each other, as soon as IgM pentamers come, they cross-link them and these RBCs clump together. This reaction is called agglutination. So IgM can react with multiple antigenic particulate matter, particles. So it agglutinates the particles. So it can agglutinate the RBCs. If there are multiple bacteria, let's suppose these are the bacteria, and you know every bacteria has multiple antigen or one antigen? Multiple antigen, and every, for example, this bacteria has 3 types of antigens and every antigen is multiple copies. So if there is IgM, so IgM will react against this, react against this, react against this. So all these are, what is happening to them? They are cross-linked. These bacteria are cross-linked. So we say bacteria are agglutinated. So we say that IgM antibody is agglutinating antibody. that if there is some antigens which are in particle forms, it will make the aggregates of particles, right? So number 1, IgM is act as B cell receptor. IgM in the blood is or serum is pentameric with decavalent, and it is agglutinating, yes, agglutinating what? Antibodies. These are agglutinating antibodies, and of course you will remember this is antibody of primary response or secondary response? Primary response. It's antibody. In primary response, IgM is the main antibodies. In secondary response, it is far less than IgG. Am I clear? Then another thing about the IgM is that IgM can activate complements, right? You know, complement proteins are produced by the liver, right? And IgM can activate the complements. There are 2 antibodies classes which can activate the complements. By that number 1 is IgG and number 2 is IgM, because IgM antibodies, these are 5 groups held together by J chain. Actually, these Fc portion of the IgM can bind the complement proteins with them, and they can activate complement. This is C1, and C1 can activate C4, C2, you know it. Then C3, C5, 6, 7, 8, 9. So this pathway of complement activation where antibody Fc portion can bind with C1, 4 and 2 and activate the complement pathway through this pathway, this is called classical complement pathway activation. What is this? Classical activation of the complements. So IgM has one more function. What is that? This is complement pathway activation. But there is one thing, the Fc portion, the receptors for Fc portion of IgM are not present on our cells. So IgM cannot bind with our cells. Let me tell you. If there is a bacteria here, how the IgM is going to kill? IgM can kill the bacteria or destroy the bacteria by activating the complements. Let's suppose here is IgM. Of course, now this group of IgM will activate the complements C1, 4, 2, C3, 5 and 6, 7, 8 and 9. You know C5b, 6, 7, 8, 9, this is called membrane attack complex. This you will study in detail in complement system activation. So C5b, 6, 7, 8, 9, this complex will puncture the membranes of the bacteria and bacteria will die, right? But IgM Fc portion cannot bind with neutrophils or macrophage cells. Why? Because the Fc portion does not have receptors on the phagocytic cells. So it cannot opsonize the bacteria. You know, opsonization is a different concept. Opsonization means that let's suppose here the bacteria, here is antibody molecule. Antibody molecule on one side bind with the bacteria, on other side antibody molecule has a receptor on a, this is a receptor for on a, suppose what is this cell? Suppose this is neutrophil or macrophage. So on the surface of neutrophil or macrophage, if there is a receptor for Fc portion of the antibody and an antibody on one side catches the bacteria, on other side binds with the neutrophil or macrophage, can now this bacteria run away? It is caught and then very effectively it will be phagocytosed. This bacteria cannot run away. Is that right? Because there is an antibody which can bind on one side with the bacteria and other side with the phagocytic cell. Right? And phagocytosis of this bacteria is facilitated in the presence of this antibody. Such antibodies are called opsonins. What are they called? Opsonin. IgM is not opsonin, because if IgM bind here, IgM cannot bind with the phagocytic cell. This is IgG. So antibodies belonging to IgG class act as opsonin, but antibody belonging to IgM class cannot act as opsonin. Is that right? So about the IgM, just important points. Number one, IgM is monomeric, right, and present on the B cell surface as B cell receptor. In the serum, IgM is present as pentameric, decavalent, large molecule, mostly in the plasma circulation, right, because they are trapped in that, not in the other body fluids in significant amount, and IgM is pentameric with decavalent molecules, and it can produce agglutination reactions. Is that right? It can activate the complement by the classical pathway. And of course, we should remember IgM is the main antibody of the primary immune response. Is that right? Do you have any question up to this? So let's have a break. So we were talking about the classes of antibodies or classes of immunoglobulins, and we have already discussed that primarily there are 5 classes of immunoglobulins. One way to remember is GAMED. GAMED. GAMED stand for what? Immunoglobulin G, Immunoglobulin A, Immunoglobulin M, Immunoglobulin E and Immunoglobulin D. Already we have discussed in detail about Immunoglobulin M, right? Now we will talk about Immunoglobulin G. Why Immunoglobulin G is called Immunoglobulin G? Because the heavy chains in the Immunoglobulin G belong to the gamma group. Is that right? Now, what are the important points related with Immunoglobulin G? Immunoglobulin G is number 1. It is the most abundant immunoglobulin in your body. Most abundant immunoglobulin. You know why? Because with repeated infections, immunoglobulin G keep on accumulating, right? You must be knowing that immunoglobulin G is the antibody of secondary, yes, response. Again, as we mentioned previously, that let's suppose this is the time, here is immunoglobulin concentration. If there is repeated exposure, this is suppose first exposure, second exposure, third exposure to the same antigen. So initially the immune response which will come, that is called primary immune response. After that there is secondary immune response, and with repeated exposure there is now another secondary immune response. Again, I will repeat, as I mentioned previously, there is a big difference in primary immune response and secondary immune responses, right? Why? Number 1, in the primary immune response, there is a slow and gradual increase in antibody concentration, and it reaches to the peak at low level and gradually it comes down to very low level. And the main antibody in the primary immune response is IgM. This is mainly IgM, right? In primary immune response, concentration of IgG is very low, right? But with every new exposure, suppose this is our second exposure. Now, when the antigen comes second time in your body, it will find some of the memory cell, right, which were activated in the previous response. And those memory cells will rapidly proliferate and give very strong antibody formation response. And now the antibody will be dominantly IgG, right? But during this time, right, after the first exposure, for example, after some time when the second exposure will occur, during this time from the bone marrow, some new B cells may have come and they will be taking the second exposure as their first. You are getting it? And they may produce what? Another IgM production. So what really happens with repeated exposure? With repeated exposure, IgM is produced with every exposure, right? But in the primary exposure, on the first exposure, IgM is the main antibody. As far as IgG is concerned, IgG is during the first exposure made in very small amount, but with repeated exposure, concentration of IgG keep on increasing. Why the concentration of IgG keep on increasing? Because the cells which originally made IgM, now they have switched, they have done the class switching and they have started making IgG. When there is repeated exposures, it means during every exposure, some plasma cells are, B cells and plasma cells are made and they initially make IgM and then switch to IgG. So when you have repeated exposure to the antigen, you have lot of B cells, memory B cells which are ready to produce lot of IgG. Is that right? Another thing which is very important to remember, half-life of IgM is short, but half-life of IgG is long. That is why if you get antigen today, you will make IgM, right? But gradually IgM will its half-life is less, so gradually its levels will go, rather not gradually, rapidly in your blood, the IgM level go down. But in repeated exposure, when you make IgG, because its half-life is longer, it will stay in your body longer time. Am I right? Now, let me mention one thing again, that what really happens, that if I get infection today, first time, then against that antigen, I will be making IgM, right? But if I got some infection 5 years back and again I get the same infection today, then I will make IgG because I have a previous exposure, and previously you can say previously trained memory cell will be reactivated and they will produce IgG, and some new B cell will also be recruited and they will produce IgM. So what really happens, it's a very important concept, that if against an antigen you have high titers of IgM in your blood, it means that infection is recent. But against another infection, if you are having only mainly IgG in your blood, then it means this is the past infection. But if you have right now in your blood against a microbe, IgG as well as IgM, it means that you had previous exposure to that organism as well as you have current exposure. Am I really clear? Right? So, what I am talking about, IgG is the most abundant antibody present in your body. Number 2, it is the antibody mainly in the secondary response, right? Thirdly, IgG is present as monomer. It is monomeric. Again, keep on comparing monomer. Keep on comparing with the IgM. Look, IgM was the third most abundant in the blood. The first most abundant is IgG, then second most abundant is IgA, and third most abundant is IgM. Again, the concentration of antibody in the blood decreases according to the sequence of alphabets in GAMED. That IgG has the highest concentration of the antibody, IgA has less than that, IgM is more or less, IgE is very less, and IgD is in traces. You get it? So, keep on comparing when we are studying IgG with the IgM. That IgG is the most abundant, IgM in the blood is less. Number 2, IgG is antibody of secondary response, but IgM is antibody of, yes, primary response. Is that right? Then, IgG is as monomer, but IgM is present in the blood as pentamer, right? IgG circulate in the blood as monomer, so we can say that it is having 2 heavy chains and 2, yes, light chains, and heavy chains belong to gamma and gamma. Is that right? Because it is monomer and it's a small molecule, so not only it circulates in the blood, this also comes into other body fluids, right? So IgM is mainly in the blood, IgM is mainly in the blood, IgG is in all body fluids, but IgA, which we'll study later, it is specially secreted in our body secretions. Is that right? IgG is not present in our body secretions in much amount, right? Again, it's because it is monomer, so it must be divalent. It must be divalent. Is that right? Now, another thing which is important about it is that it crosses placenta. It crosses placenta. So it means that on the placental cells, placental cells express the receptors for Fc portion of IgG. Let's suppose here is placenta and here is your fetus, right? Now, what really happens that the cells on the placental surface, the cells which are facing to the maternal blood, they express the receptors for Fc portion of IgG, and so IgG from the maternal side, from mother blood, IgG bind with these receptors, and once these receptors are loaded with IgG, they undergo endocytosis process, and then and through the pinocytosis and endocytosis, they are transferred from maternal blood side to the fetal blood side. So mother is giving a very precious gift to the baby. What is the gift? IgG. So IgG basically is a special maternal gift to the baby. So when baby will come to the world, he will have lot of IgG from his mother. Why IgM is not given to the baby and IgG is given? Is there any special advantage in giving IgG and not IgM?
IgM is too big.
No. Mother gives very big things to the baby. Now look, it's very simple. Let's suppose there is a Japanese mother who has come to visit America and she is living in America for few months. If she gives only IgM to her baby, she will give only those protection against those infections which she currently suffer. You are getting it? But if she gives her baby IgG, then in her blood she has IgG against almost all infections she suffered in her life. And usually baby live in the same environment in which mother lives. Fathers are a bit different thing, right? So what really happens that mothers, this is the best gift they give to their babies, that mothers have the pool of IgG, or every person has a pool of IgG antibodies directed against many microbes for which we have been exposed in our past years. You are getting it?
Yeah.
So if a mother is giving IgG to her fetus, so she is giving immunological protection to the fetus against many infections, not only against the current infection but also against her past infections. But if mother could give only IgM, then mother will protect the baby only against the current infection. Is that right? So it is a superior strategy which has evolved during evolution, that human females give to their baby transplacentally their own IgG, the pool of IgG. If a woman is living in Africa, right, she will have different pool of IgG. A woman who is living in USA, she will be exposed to different infections in her life. She will have a different pool of IgG. And woman who is living in India, she has been exposed to different set of infections, and she will have different types of IgG in her blood. So all these three mothers will give their fetuses different type of pools of IgG, which are best suited for the baby protection once they take birth. Am I clear? So there's a logic in that. Am I right? Okay, there's a very important concept. If a baby is born and baby is having IgG against the syphilis, newborn baby has IgG antibodies against the syphilis, but baby does not have any IgM against the syphilis. Do you think baby has been exposed to syphilis, treponema pallidum or not? Let me repeat a question. A newborn baby has IgG against the treponema pallidum, the causative organism of syphilis, but that newborn baby does not have IgM against the syphilis. What do you think? Baby has been exposed to syphilis organism or not? No. Probably mother was exposed to the syphilis somewhere in the past and she had anti-treponemal IgG and she passed it to the baby. But if during the pregnancy mother is suffering from syphilis during pregnancy and treponema pallidum through placental route, treponema pallidum from maternal blood goes to the baby and fetus, and baby gets the organism of syphilis. Such baby, if he takes birth, right, in such baby's blood you will find IgM directed against treponema pallidum. Am I clear? So a newborn baby, if newborn baby has preformed IgM against an organism, it means that baby was exposed to that organism during intrauterine life. Am I clear? But if a baby has IgG against multiple organisms, then it means probably these IgG, not surely these are IgG molecules are coming from maternal side. So IgG is most abundant. It is antibody of secondary response. It exists in monomer, so it is divalent, and it crosses placenta very efficiently. And how it works? Number one, it can activate complement system by classical pathway. Let me tell you what exactly is meant by this. Let's suppose here is a bacteria. Okay, let's suppose here is a bacteria, and these are bacterial antigens, and you are having IgG antibody, right? And as soon as IgG antibody bind with the antigen, its Fc portion become active, and now it can activate complement. So IgG can activate complement number 1, C1, 2, 4, 3, C3A will go away, C3B will be there, then C5B, C5A will be away, and then C6, yes, 7, 8 and 9, right? This is membrane attack complex, this much, right? So basically IgG antibody, once it binds with the antigens, then its Fc portion is activated, which can activate the complement by classical pathway and kill the microbe. This is one way. Secondly, IgG can act as opsonin. Now what is meant by opsonization and opsonin? Actually, when IgG bind with the bacterial antigens, its Fc portion become active, and neutrophils and macrophages are having membrane receptors for the active Fc portion of IgG. So if this is a neutrophil, then neutrophil will have receptor for the active portion of right. This is a neutrophil, and neutrophil had this specific receptor, and this receptor could bind with the active portion of IgG. So IgG on one side is holding the bacteria. Of course, bacteria is very sad, right? IgG is holding on one side bacteria, and other side it bind with the neutrophil. Now bacteria cannot escape, right? And these receptors give signals into phagocyte, and these signals stimulate the phagocytic process. So actually when IgG bind on the particulate matter, for example bacteria, it enhances the phagocytosis of these particulate matter or bacteria, right? This process is called opsonization. What is opsonization? Opsonization is a process in which opsonin molecule bind to a particulate matter. On one side they bind to the particulate matter, which may be bacteria or a cell. On other side they bind with the phagocytic cell, and they facilitate the process of phagocytosis, right? So IgG act as opsonin. So IgG can kill the bacteria by activation of complement, or IgG can help the neutrophil and macrophage to very effectively phagocytose the bacteria which are opsonized with the IgG molecule. Plus IgG molecule can neutralize yes, what? Neutralize virus and toxins. Now, what is the concept of neutralization of virus? Yes, Dr. Safa, you will tell me something. Have you heard of this term? There are some antibodies neutralize the viruses or toxins. Okay, let me explain. Let's suppose this is the target cell, right? And this is a specific virus. Suppose this is a virus and this virus is going to attach with this cell and push its genetic material in. Actually, on the surface of the virus, there are special type of proteins, and these proteins help the virus to bind with the target cell, right? On the target cell, there must be receptor proteins, and let's suppose this protein of the virus will bind with this cellular protein. So, virus will bind here like this, right? And when these two proteins interact, then virus will be taken in, or virus genetic material will be going in. So, first step for virus to attack our cell is that virus should develop attachment with our cell. Once virus is attached, then it will push its genetic material in. Is that right? IgG antibodies actually bind on these proteins and block them. If IgG antibodies bind with these proteins, do you think now virus can bind here? No. So we say capability of virus to bind and invade our cell has been neutralized by the presence of IgG. This is the concept of that some antibodies can neutralize virus. Of course, IgG is one of the very important. In the same way, there are some toxins, right? Let's suppose there is a toxin which has A component and it has one more component which is called B component. Many toxins which are produced by bacteria, they have A component, A for action, which produces action in the cells, toxic action in the cell, and B is for binding for the cell. So let's suppose this is receptor for the toxin. This is receptor for the toxin, and actually B component binds here, and only then when B component of the toxin bind with the receptor, only then A component will be translocated into cell, and then A component will do toxicity in the cell. Actually, some of the IgG antibodies, they bind with the B component, and when they bind with the B component, can B component bind with the receptor on the target cell, and can toxin enter into cell? So capacity of toxin in the presence of IgG against its B component, right, then capacity of the toxin to enter into cell has been neutralized. So this is how we say that this is a neutralizing antibody, because it can neutralize virus and toxins. Am I clear? Again, you must compare these functions with the IgM antibody. IgM could also activate the complement. So complement can be activated in a classical pathway by IgG as well as by IgM, right? Opsonin function of direct opsonization is done by IgG, but direct opsonization is not done by IgM, because there are no receptors on the phagocytic cell to bind with the Fc portion of IgM. So IgM cannot directly opsonize the bacteria, but IgG can directly opsonize the bacteria. Then neutralization of virus and toxins. IgG and IgM both can do this function. Another thing which is there, that IgM could do what? Agglutination. You remember IgM was as pentamer with decavalent, it could bind with multiple particulate matters. So IgM could work as a agglutinating antibody. But IgG, because it is only divalent, can it cross-link many cells or particulate matter? No. So IgG cannot do agglutination reactions. IgM can do, but IgG cannot do agglutination reaction. But IgG can be involved in precipitation reaction. Precipitation reaction mean that in a test tube you have put the antigens. There are many antigens, and then you add to this IgG. IgG and antigens may make, what is this? They may make antigen antibody complexes, and because these are small complexes, so they may settle into bottle, right? So they play a role in precipitation reaction. So IgG can play a role in precipitation reaction, but IgG cannot play a role in agglutination reaction. IgG cannot play a role in agglutination reactions. Agglutination means that antibody should cross-link multiple particulate matter, right? Like multiple bacteria are cross-linked and clumped, or multiple RBCs are clumped. This is a function of pentamers of IgM. Is that right? So these are a few words about IgG. Now we will talk about IgA. Now IgA, do you think IgA antibody, it is the second most abundant antibody in your blood. But the very special thing about IgA is that not only it is present in your blood, but in large quantities, IgA is also present on all mucosal lining of the body and in all body secretions, right? So that makes IgA very important antibody. Let me explain. Let's suppose that this is GIT and this is a GIT mucosal cell, and here is your blood circulation, and let's suppose here you have a plasma cell. Is that right? These plasma cells are producing IgA, right? Now when IgA is released, now let's suppose these are IgA antibodies. Some of the IgA which are released, they are held together by J chain. They are held together by J chain. So it means they are released as dimeric form. But some of the IgA is released as monomers. These monomers will be going to the blood. So you must remember IgA circulate in the plasma in a monomeric form. IgA circulate in the plasma as monomeric form, or this IgA is also called IgA1, right? But they are not cross-linked by the J chain. But this these two molecules of monomers of IgA which are held together by the joining chain, and this is now becoming dimer. This dimer is internalized by, it enters into, what is this cell? Mucosal epithelial cells, right? And once IgA dimers are inside, already they were joining with which chain? J chain. Then epithelial cells of mucosa will produce another protein which is called secretory component, and secretory component also bind with that. Secretory component. So secretory component is a small protein which is synthesized by mucosal cell, and this binds with the IgA dimers, and then secretory component help it to the whole complex to go to the lumen. So eventually this dimer of IgA, right, with the two joining and one, what is this? Secretory component. Whole this complex is secreted on the mucosal lining, right? These IgA are very good molecules because whenever virus or bacteria they are coming, those viruses will bind here, and then of course they cannot invade the mucosa. So many antigens which are coming to the mucosal lining, those antigens or virus or bacteria, they will bind with the pre-formed and pre-secreted IgA, and in this way they will be neutralized, they will not be able to invade in our cells. Is that right? That is why IgA is said to be antiseptic paint of our mucosal linings. This is also called that IgA molecules, dimers are antiseptic paint on our mucosal linings, right? It means, let's put it there, the important information, IgA is the second most abundant antibody in our body, right? Secondly, It is present as monomer in our, yes, it is present as monomer in our blood. It is present as dimers in, yes, dimers in mucosal surfaces plus body secretions. Number 4, that IgA, right, this is present in which mucosal membrane? Every mucosal membrane. Throughout the gastrointestinal system, it is painted with IgA. Urogenital system, it is painted with IgA. Your respiratory system, all respiratory mucosa is painted with IgA. Even your conjunctival mucosa is painted with IgA. So it's a mucosal defense mechanism against the invasion of many microbes, right? Secondly, which secretions? All the body secretions, all respiratory secretions, gastrointestinal secretion, urogenital secretions, even in the tears, in the saliva, in the milk, and especially colostrum has a large amount of IgA. Actually, this is very interesting and very important to know that when newborn baby is born, baby is having a lot of IgG molecules transferred from the placenta to protect the baby against many infections. But after the birth, if mother is breastfeeding, then baby is getting a lot of IgA from maternal milk, and this IgA will line the baby's GIT mucosa so that if some bacteria or virus come to the baby's gastrointestinal system, they will be neutralized. Is that right? But mothers who are not doing breastfeeding, their children have more often gastrointestinal infections and diarrhea, because the formula milk which are coming, they are not bringing IgA in that. Is that right? So it is in high concentration applied on the mucosal lining. It's in high concentration present in the all body secretions including milk and colostrum. And then another important thing about IgA is that it does not activate the complement by classical pathway, but it can activate the complement by alternative pathway. So classical pathway, complement activation, no. IgG or IgM could activate the classical pathway, but IgA does not activate the complement by classical pathway, but it does activate the complement by alternative pathway. It can activate the complements by alternative pathway. After this IgA, now we talk about IgD and IgE. So let's talk about IgE. IgE is the class of immunoglobulins which are having heavy chains belonging to the epsilon group, IgE. IgE antibodies produced in small amounts as compared to the IgG, IgA and IgM. Right? IgE is in the blood in very small amount. Both of them are in traces in the blood. Now IgE, once it is formed, it loves to bind with the mast cells and basophils. IgE is also called cytophilic antibody. Cytophilic antibody means it loves the cell, right? For example, once IgE is made, suppose this is IgE. Again, it has two heavy chains and two light chains held together by disulfide bond. So IgE exists as monomer with divalence. But most of the IgE right now which is present in your body, that is bound with the mast cells, because mast cells have receptors for Fc portion of IgE. But when antigen bind here, only then this Fc portion become active, stimulate the mast cells, and when mast cell is stimulated, when an allergic substance, actually when we say that, for example, if you have allergy to penicillin and you have made anti-penicillin IgE antibodies, then IgE antibodies will be loaded on the mast cells, and when allergen come bind with the IgE antibodies, actually allergen usually cross-link multiple IgE. These are the IgE antibodies which are previously formed and loaded on the mast cell receptors. When the allergen comes, suppose this is an allergen, right, it will activate, bind with the multiple IgE molecules on the mast cells, and they will come together and then they will give signals to the mast cell, and mast cell will be activated. Number one, signals will go to the membrane, and from the membrane there will be release of prostaglandins and there will be release of leukotrienes, plus there will be release of lipoxins, right, plus there will be release from the membrane of platelet activating factor. So what really happens that when IgE is activated on the surface of mast cells or basophil, basophil is circulating into blood, mast cells are the connective tissue cells present in your connective tissue throughout except central nervous system. Central nervous system does not have mast cells. So when IgE is loaded on the mast cells, it activates number one the membranes, and from the membrane lot of chemical mediators of inflammation are released like prostaglandins, leukotriene, lipoxins and platelet activating factor, plus they give signal to the cytoplasm, and these signals when they come to the cytoplasm, number one to the membrane, number two signals are going to cytoplasm, they activate the granules, and mast cell degranulation releases, yes, histamine. Do you know that? Histamine, 5-hydroxytryptamine, plus it releases proteases, proteolytic enzymes, plus it releases, yes, neutrophil chemotactic factor and eosinophil chemotactic factor. So what really happens? Once allergic substance bind with the IgE, which is already loaded on the mast cells, mast cell membrane activation releases lot of inflammatory mediators, as well as mast cell cytosolic granules release also produces chemical mediators which enhance the inflammation, and if even signal will go to the nucleus, and nucleus will lead to formation of new messenger RNA which will translate into proteins, and some proteins will be secreted out which are actually cytokines, like it will maybe releasing tumor necrotic factor or interleukin 1. So IgE plays a very important role in activation of the mast cells and basophils during type 1 hypersensitivity reactions, during which all these chemical mediators are released into tissue and produce severe inflammatory reaction. Am I clear? Then IgE not only bind with the mast cells and basophils, but IgE plays a very important role in defense against parasites, especially helminths. For example, against Scarus, strongyloids, or against hookworms, against trichinella. These are different type of parasites. People who are infested with parasites, they have very high level of IgE. IgE, which is directed against the parasite antigens that help us to kill the parasites. For example, if there is a parasite here, right, of course these are the antigens of the parasites. IgE will bind on the parasite antigens, and IgE also has its receptors on the what are these cells? This is not mast cell, this is eosinophils. These are eosinophils. So what really happens that IgE antibodies can coat the what are these parasites, and their Fc portion can activate the eosinophils, and then eosinophils will release very important destructive products, and these destructive products have one important function which is called major, yes, basic protein. And this major basic protein and some other substances which are released by the granules of the eosinophil, they destroy the parasites, right? In this way, IgE helps us to kill our parasites, right? And the people who are having these parasites in their body or the larvae of these parasites, these people have high concentration of IgE. And now we come to the last antibody group, and last class is IgD. The good news about that is we don't know much about that. This is present in the blood in very small amount in traces, and it is present on the B cells as B cell receptor. You remember B lymphocytes were having on their surface IgM, but there are also some IgD. So IgM and IgD are present in monomeric form, expressed on the surface of the B lymphocytes, and they act as receptors for the antigens. So B cell receptor or BCRs are mainly IgM, and some of them are IgD. Is that right? So, okay, I will ask you some questions. Now we will talk about another way to type the antibodies. Antibodies can be classified as idiotypes, allotypes and isotypes. Another way to classify antibody is allotypes, isotypes and idiotypes. Now listen, let's suppose here you are. And from your circulatory system, we remove all the antibodies. We remove all the immunoglobulins. Now all the antibodies which we can bring out from one person, they have something common. Right? Because antibodies are what? Antibodies are just peptide chains. These are 4 peptide chains, 2 heavy chains and 2 light chains held together by disulfide bond. Is that right? And antibodies, because they are protein, so they are antigenic. For example, if your antibodies are injected into a donkey, suppose this is a donkey. I don't know how to draw a donkey, but you can imagine it. This is a donkey, right? And if human antibodies, suppose antibodies from this person are injected into donkey, these are the antibodies. When antibodies will come, do you think for donkey these antibodies are self proteins or foreign protein? It depends on the person actually. Right, human antibodies for the donkey are foreign proteins. So donkey's immune system will take these human antibodies as antigens, and donkey's immune system will produce antibodies against human antibodies. You are getting it? So it means human antibodies are antigen because they are proteins, they have antigenic areas, or we call them, they have antigenic determinants. Is that right? Now, actually, they found that all the antibodies which are present, removed from one person, all the antibodies which are present in one person, they are having something common. For example, some amino acid sequence here in the light chain and some amino acid sequence, let's suppose here. This amino acid sequence is present in all the antibodies of this person, right? So, according to this concept that this person has inherited a special type of alleles, and those special inherited alleles, all the antibodies from this person have some amino acid sequences which are common, right? And according to these amino acid sequences, antibodies vary from person to person. For example, in my case, in my all antibodies, suppose here is histidine and here is leucine. Maybe in your case, at this point, there is histidine and threonine. So my all antibodies have histidine and leucine, and your all antibodies have, suppose, histidine and threonine. So it means all my antibodies have same antigenic determinant here, and all your antibodies have a different antigenic determinant here. But for your antibodies, all of them will have the same antigenic determinant. So this, if we classify human antibodies according to this antigenic determinant, we say we are doing allotyping. So I have one allotype and you have one allotype. So all the antibodies which are present in one person, that is determining its one allotype. Is that right? Opposite to that, isotype means the differences in antibodies in the same person due to heavy chains. You know the constant of the heavy chain. These are the constant region of heavy chain. How many different types of heavy chain one person is making mainly? 5 types. Because these are the 5 basic classes. So we can say if we bring all his antibodies out and try to classify his antibody according to constant of the heavy chains, how many will be main group? 5. So if donkey is going to make antibodies, against the constants of the heavy chains, how many types of antibodies you will make? 5 types. Is that right? So this, when you classify antibodies in a person according to the constants of the heavy chain, they are divided into 5 groups, and those groups are called isotypes. What are they called? Isotypes. So I have how many types of isotypes? 5. And you have how many types of isotype? 5. And my isotype and your isotypes are same. This is very important. You have IgG, IgM, IgD, IgE, IgA. I have also same groups. Is that right? But donkey does not have the same group. For donkey will make anti-isotype antibodies. When you put my antibodies into a donkey will make how many types of anti-isotype antibodies? 5. But donkey will make how many types of anti-allotype? 1. I have only one allotype. So if you inject my antibodies into a donkey, the donkey will make how many anti-allotypes? 1. Donkey will make how many anti-isotype? 5. So one person has how many isotypes? 5. Or one person antibodies, when they are classified according to allotyping, in one person how many allotypes are present? 1. If one person's all antibodies are classified according to the isotypes, how many groups are there? 5. Thank God you know it. Now ideotype. Ideo means unique. Actually, if I have been exposed to thousands and thousands of many different virus and bacteria, then it means I've made thousands and thousands of different type of antibodies with specificity for different antigens. Is that right? And why now one antigen is, let's suppose one antibody, you know, every antibody has different, there's special variable areas here, variable area of the heavy chain and variable area of the light chain, so that this antibody molecule must bind with a specific antigen. Now tell me one thing. In one person, if you try to classify all my antibodies according to these variable regions, how many antibodies groups I will have? Millions. Right? This is say that all the antibodies which are made by one clone of B cells, they have the same variable region. But actually, every person has capacity to make millions of different antibodies directed against different antigens. So it means in every group of every antibody, these variable regions are so unique that every antibody will bind with a unique antigen. Or to be more clear, all the antibodies which are derived from one clone of B cell, they are directed against a unique antigen, and all those antibodies will have a unique amino acid sequence here. Is that right? Now, if you remove my all antibodies and inject into donkey, and if donkey is going to make antibodies against variable regions of my antibodies. He will make millions of different type of antibodies. We say that he is making anti-idiotypes. Idiotypes. So what is idiotyping? Idiotyping is the typing of the antibodies according to their variable regions. Isotyping is the typing of the antibodies according to the constant of the heavy chain. Allotype is typing of the antibodies from person to person, right? So because every antibody which is directed against different antigen has a different type of idiotype. So what are idiotype? Idiotypes are the antigenic determinants made by the amino acid sequence in variable region. Let me repeat it, it's worth repeating. What is idiotype? Idiotype is the antigenic determinant made by the amino acid sequence present in variable part of the antibodies. And one person may have millions of idiotypes. Then isotype. What is isotype? Isotypes are antigenic determinant in the antibodies of a person which depends on the amino acid sequence in constant of the heavy chain. Is that right? And allotype is the antigenic determinant present in a person's antibody according to the allelic morphology, right? Again amino acids. So I can say that all my antibodies will have one point unique, which is unique to my all antibodies, right? So as far as this is allotype, so how many allotypes I must have? 1. How many isotypes I must have? 5. How many idiotypes I must have? Millions. Is that right? So when we say that there is anti-idiotype antibody, it means that antibody will react here. For example, if donkey make anti-idiotype, that will react here. If donkey make anti-isotype, that will react here. If donkey make anti-allotype, that will react here. Is that right? Any question? Right? Class dismissed.